Well, it's a pleasure to be here and share a few things that my research group has been working on. We have a new water program at Duke. We hired 5 in the hydrology area, and we looked at some of the things that Waterloo was doing and hired some people in the groundwater area and streams and all kinds of areas, so we're trying to catch up there. But I've been working on wetlands, which are 80% water, so essentially wetland students, I tell them you can walk on water, and that's a pretty good actually aspect of the program. Today I'm going to talk about decoding the secrets of carbon preservation in peatlands along a boreal to a tropical gradient from Minnesota to Peru. We actually do have a few samples in here from Canada. And I have to thank my co-authors, my research associates, Dr. Flanagan and Wong, who have been with me for a number of years. But Sue Hodgkins and Jian Qian, she was a postdoc that went back to China. Dr. Ho is still with me, and Dr. Cooper and Dr. Chanton are both at Florida State. They've been doing some of the high-powered chemistry. So the real tough questions, you'll have to phone in to them to see what's going on. So hopefully this will work. So if we take a look at this gradient, the regional gradient, we're starting at the top. We're basically going all the way here from Minnesota all the way down to Peru, and we're looking at these are all bogs I'm going to be studying, and you'll see what the question is in a minute. But essentially, we have the boreal one from Minnesota. We have a temperate subtropical one in North Carolina, which are called pocosins, which means swamp on a hill. We have the subtropical Florida Everglades, which I worked in for about 15 years. And then we have some bogs in Peru. So the basic hypothesis that our group's been working on for about 10 years is, and I'll go through this in the talk, we'll basically try to cover some of these. Not all of these are answered for sure, and a lot of them have more questions at the end, but our basic hypothesis was high phenolics and black carbon peats, which are basically derived from some like fires, derived from native fire-adapted high phenolic plant species, generates a dual protection mechanism in peatlands to enhance carbon sequestration and reduce carbon decomposition. I'm going to show you today what happens to your peats when they come in contact with our peats. 2, high phenolic contents can improve the natural resilience of peat, decreasing its sensitivity to drought and global warming, as well as drainage. 3, increases in black carbon from low-intensity fires can protect peat from decomposition and reduce greenhouse gas losses. Fire in peatlands is anathema because everyone talks about preventing fires because deep peat fires are a disaster like they have in Indonesia, Canada, Alaska, even in our country, North Carolina. And 4, the importance of the dual resistance mechanism, high phenolics and black carbon increases in peat near the tropics. So basically the supposition was that why should tropical peatlands even exist under high temperatures, more drought, and conditions that really, if you put your Canadian peats or Minnesota peats in, would disappear quickly, but they don't. So we came up with this dual latch hypothesis that subtropical and tropical fire-adapted trees and shrub communities produce these higher phenolics and aromatics, which I will explain what they are in a minute if you're not familiar with them. And low-intensity fires produce higher black carbon aromatics than found in northern sphagnum carex communities. Notice I said sphagnum carex, I didn't say shrub. Low-latitude peatland decomposition is downregulated by higher production of these compounds than found in your northern systems. So if you look at it in cross-section, or what is sort of my cartoon here, basically this system works the following way. Native species, and that's the critical, this is a plant-derived protection mechanism, produces peat soil quality with those with higher aromatics and phenolics and black carbon affect the enzymes and the microbes, which I'll show you. I'm not saying that flooding, the hydrology, which is in the lower right-hand corner, or that fire have dramatic effects on peatlands. We all know they do. I'm just simply saying we haven't looked at why some peats are more resistant than others, and it has to do with this, what we think is this dual control mechanism. Now, the first piece of evidence we looked at was the paper we published in Nature Climate Change with Dr. Wang and myself and Dr. Ho, was basically looking at, is there a difference? And if you look in the sphagnum, the herbs, the shrubs, and the trees, you'll notice that the shrubs by far have the most phenolic material. Phenolics, phenol, basically you can buy a throat spray that will kill bacteria. If you buy a chloroseptic in the green bottle, not the red or blue, you'll get there where you only get benzocaine, which makes your throat feel good. The 2% phenol will actually kill bacteria. And so it's antimicrobial and it's antifungal. So if you look, you'll also see that the Pocosin systems in the green are off the charts compared to the Boreal peatlands. The Boreal peatland data is from about 57 different bogs, mostly from Canada. So we have this high resistance group, which is a shrub group. And so then we went actually, and this is our early work, we'll build up to the 4 sites. This is the total phenolics in milligrams. This is in the sphagnum, the herbs, the shrubs, and the peat. And I should say, you should have noticed in the other one, sphagnum, peat, virtually has no phenolics. They have other things, cocodilic acids and other things, but they don't have phenolics. And here you see from the Boreal peatlands from Minnesota in the black, and then you can see the Pocosin shrubs here again, and then you can see the Everglades, the Pocosins, and the Everglades sit really high, especially the shrubs, in terms of this as we're going. At this time, we didn't have any data from Peru, which was a little hard to get into, and also very hard to get the samples back into the US. You think we have an immigration problem, try getting peat samples back across the border. So if you look at these phenolics, there's a paper that came out, it's a really interesting paper just to show you, if you look at bogs, people never thought about this. We know about the electron transport system, and we know about oxidation redox reactions. And so we know that oxygen is the most commonly used, then nitrate, and you go down the line with iron and iron reduction. But it's now been shown through a number of recent papers that this dissolved organic matter, basically under anaerobic conditions, you actually have redox active moieties. You have electron donating phenols, which you see in this picture, this part up here. And you actually have quinones, electron accepting components. And so on the right-hand side, you should actually see that in these peatlands with 80%, 70% organic matter, hardly any other minerals in the redox chain, you actually have an organic phenol oxidation quinone reduction system that actually undergoes oxidation reduction. So they play a very important role. As you know, you can't decompose peat unless the microbes can use it, and you have a transfer of electrons. So the first piece of data that we put together was, does phenolics actually inhibit CO2 respiration? We did this on our Pocosin systems. I'm not showing you that figure today, it's another study, but virtually we have no methane coming out of Pocosins. We have no nitrous oxide. We basically have CO2. So here is a picture of, on the x-axis, as we increase the amount of phenolics in the soil, on the x-axis here, and you look at the CO2 production, as we increase this, you see there's a dramatic decrease in the amount of CO2 production. So it looks pretty clear that in fact, in terms of greenhouse gases, and the main one, it's about 95% of all the gas that leaves that system, that phenolics are having a pretty important impact. But this is probably why the paper got published. It was an experiment I made Dr. Wong do, and that is, you always have a problem of publishing anything that's a one-off system. So essentially, we talked about what would happen if we did an experiment, sort of a cross experiment, and in this case, these are samples from Mer Bleu, actually in the Quebec area, from the bog, and we actually took, and you'll see on the lower axis here, this is Pocosin peats and Pocosin extracts, and the PS is Pocosin with the shrub extracts. I'm sorry, I'm doing this backwards here. This is, the Pocosins, and the S stands for the sphagnum. So what we really did is, if you look at this, when you had Pocosins with Pocosin extracts, you had very low CO2 emissions. When you had Pocosins and you took the Canadian Mer Bleu, you see here the CO2 level was very low. However, when you took the sphagnum peats from Canada and you actually mixed in the Pocosin soil pore water extracts, we reduced it by over 50%. And when you just had sphagnum and sphagnum, you can see this number. So essentially what happened is the North Carolina picosin extracts shut down decomposition in the Canadian peats that we tested, which was pretty cool, I think. And in fact, I should just a little sidelight, there's Freeman from Wales University has actually got a project now in Canada. He's actually adding phenolics to bogs in the eastern part of Canada to see if he can shut down greenhouse gas production. This is an idea that where you'd actually do some management on restoration. Can you add phenolics and do that? This is another experiment that was done. It was the North Carolina shrub picosin peat extract effects on the Minnesota Boreal sphagnum methane emissions. And again, you can see the same type of an experiment, except now we're looking at methane and putting them in these incubations for a long time. And you can see again here on the corner where you have sphagnum with sphagnum extracts. Again, you're getting the most methane. When you have sphagnum and you mix in here with the picosin extracts, we shut it down by almost 50%, and then you can see the picosins themselves, and the sphagnum has no effect. So we're shutting down both CO2 and also methane. Then we have this particular effect here. We see something else that's interesting, because these systems dry out, as all wetlands do when we have dry periods. And here you see the pulse effect of re-wetting. And what I want you to see and note here is on the top is that you'll notice that after we re-wet these after a certain period, that they'd been dried for 60 days and then re-wet them. You will notice that in the blue, the sphagnum peat basically took up a lot of moisture, whereas the picosin peats essentially still, the soil moisture stays fairly close. And that's because there's a lot of pore space in the sphagnum peat. Ours is woody peat, and it's almost like axle grease in places except for the very top. So there's not a lot of pore space for water. It doesn't dry out as fast, and it doesn't re-wet. But the bottom part of that story is drought. And so essentially here, if you basically dry it down, you can see that drought increases soil respiration in the sphagnum, which is in the blue at the top. And you can see you get a dramatic increase here after it gets drier and drier of your CO2, but you'll notice that the picosin peats stay pretty low all the way through that. So they seem to have built in some kind of mechanism that doesn't allow them to decompose as fast. Now we're going to go back and we're going to start looking at the gradient. That was the basis of a lot of this. Now we're going to look at both the Minnesota and the North Carolina systems. And on the upper left hand, you're going to look at North Carolina, and you're going to see this green bar. And this green bar has to do with the relative abundance. And these are dominated by fungi. There are bacteria, but we're going to talk mostly about the main decomposer. And you can see here it says shrub peats, and there's a big green, it's all green. It turns out that our systems are dominated by one fungi that's an ancient one, and most systems around the world, forests, wherever you have it, they find it at about 1%. We're finding it at 75%. It's the dominant decomposer. And if you go and look at the Minnesota peats, you will see that in fact you have a little bit of the green, but you have these blue. What you have, you put these in categories, is if you look at the relative abundance, on the x-axis up here, you see as the phenolics increase, you'll see a change. You have the slow-growing fungi increasing, and you'll see the fast-decomposing, fast-growing decreasing. And so the question is, are the slow-going there because they can deal with this nasty, complex, aromatic phenolics? Or are they able to use it, or are they the only ones that can survive? And the other part of this story, this is being submitted to Science. It should be submitted this week. And then we notice also when you do a temperature experiment, because temperature affects these microbes and how fast they go, that if you do this temperature experiment, you will see in the red here that the sphagnum peat follows this type of curve. And you can see, of course, the picosin peats are much lower. They simply don't respond to higher temperatures. We get temperatures 35, 37 degrees centigrade on the top of some of these peat systems in the suns of North Carolina. Now, going back to why and what's there just briefly, if you look on the left-hand bar, you'll see the shrubs of North Carolina, and you'll see this, I changed it to red. This is the, I don't pronounce these that well, but we can try, Archaeorhizomyces. That is that ancient group of slow-growing fungi fungal ecologist at Duke was running down the hallway hollering "Eureka." I've never seen him do that, saying, "I've never seen this amount," and we did it 5 different locations and like 10 different times, and that is the dominant group, fungal group, and it's all slow growing. If you look at the sphagnum, you can see it's made up of a bunch of, it has some slow growing, but it has fast growing, and it has some unknowns. Quite a different array, and we've got a lot of information on that, but that's not the main focus of the story today. It's just to say that there is probably a reason why with those aromatics we're getting that slow growth. Now let's look at that gradient and try to put the picture together and finish up some of this. So we'll go back and look at, this is what they look like, starting at the top. Up here, you can see some pictures from the Minnesota Marcel site, and that is the spruce site. That's the largest warming experiment site in the world. It's a $25 million a year site. There's about 30 universities working on it. That's where DOE is putting all of its money. So you can see what those sites look like. The next one is the North Carolina Pocosons, and you can see what they look like. They're shrub bogs. That's the Pocosan word, basically, means swamp on a hill, which was an Algonquin Indian word, but they are interesting peat systems. Of course, the Florida Loxahatchee system, that's the bog part of the Everglades. Most of it's a fen, but they have tree islands and open water and carricks. And then the tropical Peru, Nueva York is basically a tropical pole forest bog. So what that looks like, because you've seen most of the rest, and we took some pictures. This is the trees are dominated here, Platycarpum and Pachyra. This is what it looks like. And this is another site we went to, Miraflores, very similar vegetation. It's all woody peat. And so now we take a look at this peat, and before we get into the chemistry of it, you can see in the upper left, this is the peat from the sphagnum from Minnesota, and then we have the Pocosan peat, and then all here we have Everglades, and then we have Miraflores in Peru, taken mostly with the Russian core or our box core. So just to give you a little picture of what that's like, we look then across this, what's the percentage of carbon? Surprisingly, the Minnesota sphagnum peat has got the lowest amount of carbon, and the rest of them are all sitting above 50, 60%. In fact, if you look at it and you do the stats on it, you can see that Minnesota sits below the other two, but basically the rest are pretty much the same. If you look at the nitrogen, just to see some of these other factors that could affect the microbes and the decomposition, Minnesota and North Carolina are pretty poor in nitrogen. Florida is a little bit higher. In fact, it's because of the blue-green algae that grow there. You can find soils in the Everglades that are 3% by weight nitrogen. So when they use it for farmland, they don't have to fertilize that. And Peru is a little bit above the North Carolina. Phosphorus is a little shocking because Minnesota and North Carolina and Florida were pretty much the same, but Peru, for reasons, and I have no idea why, obviously maybe due to some of the minerals coming in there from the upland area, but these are bogs. A lot of this is, rainfall doesn't have much. I don't have an explanation why that's a little bit higher. And then let's look at phenolics. And so we can look at phenolics. It's not a perfect gradient, but you can see Minnesota's the lowest. North Carolina has by far the highest range here. Florida is a little bit lower, but one of the sites, these are more of a grasses. This is a shrub site, so it's more in range, and these, of course, are shrubs. So the secret of that is these are shrub, woody, lignin-dominated systems that have more of that. And if you array these out with look at these in sort of vector space, you can actually see the North Carolina Pocosans here in red, and these dots represent all the different samples that were taken. The other three sites sort of overlap in this, but the Pocosans sit out here. There's a little bit of overlap, and this is, the range on this is so wide, it has to do with, as you'll see, the time from fire. There are some fires in some of these other ones, but basically the age since the last fire has an effect on the amount. And you'll see the vector, you got phenolics over here, moisture over here, some of the major factors that are separating these out on the PCA analysis, the amount of nitrogen and DOC, but that gives you some idea where they sit in vector space. Now we get into a little bit more of the chemistry, and this is some FTIR work, and this essentially shows you when you do this at the top, you've got cellulose starting way over here, and the aliphatics here, and then you've got aromatics here with a double CO chain, and then you've got more aromatics, and then you've got the more soluble polysaccharides. And you can see Peru and then essentially Florida and North Carolina. So the peaks, that's hard to discern what those really mean looking at it like that. So when you look at some of these indexes, which are ratios of some of these, which I can go to, the thing that's really interesting is you can see from Minnesota to North Carolina to Florida, Peru, this is a humification. These are more humified. These are more resistant. More of the carbohydrates are gone, which I'll show you later. This has to do with the hydrophobicity, which is re-wetting, how they actually can respond to re-wetting. And essentially, you can see that Peru sits up a little bit at the top here in terms of its ability for re-wetting resistance. So one other way of looking at this was just finish this. This is just a draft. That's why it's a dotted line. We haven't finished all the samples yet. But if we looked at our gradient story, it isn't a perfect gradient, but you can see, again, phenolics all the way up here to 800. And the soil respiration, you can see Minnesota, Florida, Peru, and North Carolina. Whether it's basically a north to south trend, but just basically a sphagnum to shrub trend, if you want to put it that way. So one other thing, we went to another site in Los Amigos in Peru. This is a palm forest. And just to show you that I actually did go there, I'm standing there looking, smiling. This was probably my last foray into hacking away through 2 kilometers of swamp. We had to go down a rope and to get to this system. And I got in there for the first 2 days. And the third day I got back to the base at almost at nighttime. I told my grad student and another fellow, I can't, I'm leaving my pack here, coming back tomorrow. And they said, well, you only got to climb back out. I said, you don't understand. I either climb back out myself or I stay here with the pack all night. So, I basically, they said, what's going to happen? I said, I'm going to sort the samples the next 2 days, put them in the computer, and you guys are going to do the rest. So, anyways, it was what my wife was finally telling me, that I just got to stop being 25 years old again, I guess. So that's what we need grad students for. I mean, those are perfect. Strong backs. I didn't say weak minds, I said strong backs. So here we see some data that one of my PhD students, Scott Winton, and from that study, and we basically looked at 3 sites in that palm forest. And here you see the amount of phenolics in red, and we got the basin periphery, which is close to that steep slope, the basin interior, and then the flats. And you'll basically see the amount of methane being generated does basically decrease as we get to those sites where the peat has been formed by vegetation with more of that. So just again, just kind of supporting that concept. Now this is a diagram I want to put in there because, again, we've got a few sites, but I want to throw this one in because this is actually produced by Susan Hodgkins, who's one of the authors on this talk. And this is her study, which should be published, I hope, in Science, Nature, very soon. It's been in 3 reviews. And she's got far more sites than we have. And these are scientists from around the world. There's about 40 authors on this. But you can essentially see, if you look at this profile here, we've got Sweden, the Palso Swedish bogs. We've got Minnesota sites that we just talked about. We've got North Carolina pocosins, different ages. We've got the Florida Loxahatchee. We also have sites that she has from Panama, and then way out in Brunei, way out in the Indonesian area, in that particular part of the world. And you'll notice that the gray and the pink bars differ. The aromatics, which are the resistance, as you go towards the tropical area, are increasing, and you'll notice the carbohydrates are decreasing. So the idea is, this is why some of that peat is so resistant. The carbohydrates are gone from those systems, and you're basically left with some pretty nasty, more difficult things to break down. So, we're getting back to the end of this part. Now, we talked all about those aromatics. Is there other ways? The second part of the hypothesis, what about the effects of low-intensity fires? So we go back to the pocosins because we could only burn certain areas that were being burned by the fish and wildlife. And so now we look at these pocosin systems. This is a picture of the tall pocosins and the short pocosins. This is on about 3 meters of peat. This is on 1 meter of peat. And essentially they're about 9,000 years old, high summer temperatures, and the water table drops about 1 meter every summer. So the water table is way below And the fire return intervals can be anywhere from 1 to 3 to 1 to 6 years, whereas up in the north you get much longer periods, up to 100 years. So we actually piggybacked on a study they were doing where they were actually going to burn some of the fuel to keep it down so they wouldn't all catch fire. And so you see a picture A, you see the prescribed burn area, then the B, it's on fire. We had sensors out there to see what temperatures and what it did to the moisture, and then you see the C picture, you can see at the bottom what it looks like, frost profile, and then you can actually see the surface leaves. It wasn't that bad. They burn when there's only a certain moisture level, otherwise the whole thing would catch on fire. So that's what it looks like, is crispy leaves on the system. So we look at the fire effects now. If you look at the fire effects, we sent some samples off to Oak Ridge, and they did pyrolysis GCMS on this, and they actually produced a whole bunch of these nasty compounds. Many of them, which I don't have the name on here, but things like 2,5-dimethyloxyethylbenzene, some phenols, some uracilic acid. These are things that microbes don't like. If you then take a look at what we did in the lab, we actually set up some fires, and we took some of the main dominant vegetation. And this is a little complicated diagram, but essentially we're using a shrub, Lyonia. And if you look at these upper peaks here, we've got polysaccharides on this side now, and aromatics and fats and cellulose. And you look at the dash line and you look at the red dash line, these two are here, you can see a very light fire had very little effect on those. But a big hot fire burned those down. So obviously we're able to see, and we burned these at 250 degrees and then 400 degrees so that we could see what would it do to these various compounds. And a really hot fire, of course, burns off a lot of those materials. And some of the surface peats actually show that, in fact, that they've lost some of those materials from some of those fires, which meant they were hotter fires. So if you look at that, this is an FTIR-MS diagram. It shows the effective fire on soil organic matter from North Carolina, where the recent and 5-year fires, prescribed fires, were. And what you're looking at, these are hydrogen to carbon ratios and oxygen to carbon ratios. If you look at these, the lower, this reds and oranges, the lower this ratio is, essentially you get more condensed hydrocarbons. Those are the more recalcitrant ones. And so essentially you're looking at prescribed burn versus an unburned system. And this is the burn, and you can see they're getting lower numbers on the burn site, much lower numbers in the red area. And then we took some of the material in situ decomposition rates and we burned it, and we burned a bunch of twigs, and then we basically left some, and after a year you can see that the burned material that was there decomposed at a much lower rate than the unburned material. So this biochar on the outside, we call it hydrochar, actually protected that. And then finally on this part, to wrap some of this up, if we look at the light fire reduction, we've done some burns and did some modeling on this. So we now compare North Carolina, Minnesota, and Florida, and we can see if we start up here, burn versus unburned. The burn is in the red lines, and the number of days, and what this diagrams are trying to show for those areas is that initially you get a little bit more CO2 production, gas production when you have a burn. You get a burst, you'd expect that. But after a period of time, which is about a year, you get a crossover, and the burn sites decompose from that period forward. We've now done it for longer periods, 2 years, 3 years. They just don't decompose as fast. And so essentially you lose this initial amount, even with the Florida ones, but some of these take up to 2 years for the crossover point. So we've been able to develop some models to develop how long it would take for that to basically balance out and how much carbon you could store with a light fire versus not doing that. The other aspect on this very complicated diagram is that the burning, the DNL, this is the natural site in the green over here, way over to the left, that's not been burned in about 25 years. And so if you look at these various stages here, we have in the blue a 5-year fire, in the light blue a 2-year fire, and then the yellow 2 months before the 2-month fire, and then the orange before the fire. So if you compare the yellow and the oranges, different depths, but if you stay over here, the trend's pretty much the same. That essentially you get a burst, you get an increase in the diversity of fungi essentially 2 months after the fire, and then basically it shuts down. So the fire has an effect on the communities. We're still looking at that. That plays a big role, and that's true through the depths. So this is basically pretty much the end of it. So findings to date. Our research supports the dual control hypothesis. Low latitude peatland carbon decomposition is downregulated by higher production of phenolics and aromatics than found in your northern sphagnum carex communities. Higher concentrations of phenolics and aromatic compounds equal higher recalcitrant peat, which inhibits the microbial fungal activity. We've got another study, it also decreases the microbial activity of bacteria. And then finally, recurring low intensity fires equals increased aromatic carbon compounds, lower carbohydrates, and decreased losses to microbial respiration. So I'd have to thank, this has been supported by the Department of Energy. They're still one that's allowed to put the word climate in their webpage, unlike EPA. Funding from the Fish and Wildlife Service, which started this originally. And of course, thank my 2 valiant technicians who'd spend many, many hours along with grad students doing a lot of this work in the lab. So I'll leave you with 2 pictures for questions. That's actually the swamps, pole swamps and palm swamps in Peru, which are the ones that really took my steam away, as I would say. The problem was the guy way in the back, this is Dr. Flanagan here, and this is Dr. Winton, my grad student, and then this is a soccer player from Peru we hired to carry all of our stuff and get us in there. And he said he knew exactly where we were to go, and he let us off for we spent a whole day macheting our way in the wrong direction, which didn't help, but he did get to carry all the samples out for us. So the last point here is this, and the last thing, if you don't protect these systems and you don't reflood them, this is a picture of the Pecosins in 2008 that burned, and this fire burned 16,000 hectares, and it released, because we had LiDAR data, you can see it's 2.5 meters of peat. All you see is the stumps left here. Look at the size of that old cypress tree from 200 years ago. It released enough carbon in that fire to match all the federal gasoline used by federal vehicles in the United States in 1 year, 9.9 teragrams. When you say 9.9 teragrams to politicians, as Barry was mentioning, you have to get to their eyes glaze over and they go, great, goodbye, thank you. But if you say it matches all the gasoline used by all the federal vehicles in the United States in 1 year in one fire, they go, whoa. Hence, they released $500,000 to Fish and Wildlife to get this funded started. So thank you.
So, well, that was great, Kurt. So, first of all, that photograph, it burned 2.5 meters of peat in one fire. Where the hell was the water table?
This was drained. The problem was, the reason this happened is all this was going to be put into farmland, and they drained huge amounts of this to put it into farmland. And so the water table was way, way, way below. In fact, it burned right up to the 5,000 acres they had restored and stopped. But it burned 16,000 hectares right up. In fact, they thought they were going to lose everything. The farmers thought they would lose everything. They were reflooding the canals, and they were pumping water from the lakes. It took them 2 months to put this fire out. The smoke went all the way to the mountains of North Carolina. I woke up one morning and I said, it was like June, and I said, July, and I said, that's a peat fire, because I'd smelled them in the Everglades. And my wife said, no, somebody's probably got a fire. I said, it's 90 degrees. Nobody's got a fire in their house. That's peat. And sure enough, you could see the satellite photo I could show you. It goes all the way across the state. They just couldn't put it out. And that's because they had drained it. The question is how much the microbial or fungal diversity has to do versus the plants that are there. Is that?
Yeah. I mean, the plants are also responding to the fire and changing their chemistry in response.
Well, they are. But I'm saying that I guess is the chicken or the egg question. Is it are the fungi there because of the plants, or are the plants basically result what they put out and make the peat, was what the fungi and bacteria can survive in, and so. You work a lot more on that than I do in terms of that, but I would say that the plant material, they are the ones that produce the soil, the plants. So I would say that they basically control what can survive there, especially in those conditions. But you might have a different opinion on that.
No, I mean, I had looked at some similar things and found, obviously the largest effective fire or the largest impact on decomposition is the fire itself, but I didn't look at all of the fungal community. So it's a really interesting component in comparing the chemistry of the litter itself, trying to, I guess, understand how those two factors interact to influence decomposition, right?
Well, one of the things to do is, I did it many, many years ago on a different type of thing, was I basically went to the medical field. I didn't know much about bacteria or fungi or anything at that time, and you could still do that here, and that is use various, like things like streptomyces, all kinds of aspects, and knock out various groups, both bacteria and fungi, and see, you know, whether or not they would exist or could exist. And you can also do inoculations the other way and see what they would do. But that's getting beyond the realm of where we are. We just basically found out that we just had this one group that was considered ancient, slow growing, which was pretty bizarre.
Yeah, very, very weird.
Very weird. Eureka, weird. So didn't you think that we've now made a case where you have these weak sphagnum peat bogs up here? You need to import phenolics.
Well, no.
No?
Because the question I had for you is, if the plants anywhere, but you're telling us that the phenolics are much higher content in tropical, subtropical peats than us here, up here in the temperate zone. But if there were plants that didn't have phenolics, then what would you imagine would happen if the phenolics weren't there?
In the tropics?
Yeah.
Well, we have some areas in the tropics that are they're not bogs. They're basically swamp palm forests, and that was why I showed you one of them right there. And their actual decomposition rate is higher, and so some of the palms don't have very much of that. And they actually, the reason they do exist there is they're right on the river floodplain, and they flood a lot, where they don't get as dry as some of the other ones. I might also mention that. I mentioned that study that's being done in Canada, bringing phenolics into some of the bogs to slow it down. One of the aspects that we've been throwing around, and one of the aspects is that if we do have as much climate change in the north and we have drying out and we lose sphagnum, which you can do, and a lot of the areas are being invaded by shrubs. And your shrubs up here do have phenolics. And so the one aspect of that might be that actually you won't get as much greenhouse gas out of some of these when they move into the shrub category. They're starting to find some of that in Finland, a little bit in England, that where some of the sphagnum areas are dominated now by these shrubs, the surface peats are more resistant than they were below. So it's that's kind of interesting. So it isn't all bad, but some people may not want to lose sphagnum. Water is the key, though. You all know that. As you've got to keep water on these systems. If they dry out, you see the result.
So it's not all bad then, as far as climate change is concerned, and the phenolics are going to protect.
They won't save the day, but they might slow it down.
Oh, okay.
I can't save it. It's a tiny piece of the puzzle. Thank you.
All right. Well, thank you very much.